Hello, everyone. My name is Valeria Zapata, and the title of our research is Identification of Lipid Metabolism-Associated Genes in the Gliovascular Unit of the Hypothalamus using scRNA-seq in the 3xDG AD mouse model. Alzheimer's disease, the most common form of dementia, is marked by beta-amyloid plaques and neurofibrillary tangles that disrupt the blood-brain barrier. This disruption activates astrocytes and microglia, leading to the release of pro-inflammatory cytokines and reactive oxygen species, which drive neurodegeneration and synaptic loss, ultimately causing memory loss and cognitive decline. Alzheimer's has two forms, familial and sporadic. Familial Alzheimer's is inherited through mutations in APP, PSEN1, and PSEN2, impacting beta-amyloid processing and lipid metabolism, leading to abnormal lipid buildup in the brain. Sporadic Alzheimer, with a multifactorial origin, has APO4, a major lipid metabolism factor, as its main risk factor. While familial Alzheimer is less common, its defined genetics allow for controlled experiments that help identify mechanisms relevant to sporadic Alzheimer. While Alzheimer's research has mainly focused on the cortex and hippocampus, recent studies highlight the hypothalamus, which regulates essential autonomic functions and links brain and body metabolism, as also affected by the disease. Tau and beta amyloid aggregates, along with accumulating neutral lipids like triglycerides and cholesterol, underscore the role of lipids in Alzheimer's pathology. Hyperreactive astrocytes and microglia in the gliovascular unit show intracellular lipid buildup before neurodegeneration, pointing to lipid alterations in the hypothalamus as key to understanding Alzheimer's progression. Our hypothesis suggests that early lipid changes in the hypothalamus of Alzheimer's model mice are linked to differential gene expression associated with lipid metabolism in gliovascular unit cells. To test this, we aim to identify gliovascular cells in the hypothalamus of 3xDG AD mice and controls, determine the differentially expressed genes, related lipid metabolism processes, and the transcription factors regulating these genes for our experimental design. We conducted a comparison of 4-month-old mouse models to study the hypothalamus in the context of Alzheimer's disease. On the left, the control model is a knock-in mouse with a presenilin 1M146V mutation, with 4 individuals in the group. On the right, the Alzheimer's model is a triple transgenic mouse with mutations in APP, presenilin 1M146V, and tau, P301L, also with 4 individuals in the group. In collaboration with the University of Sherbrooke, we performed hypothalamus dissection and cell preparation. Then, single-cell isolation, cDNA amplification, and library construction were carried out. Subsequently, all cells were sequenced in a single pool using NovaSeq 6000, and data were acquired for analysis. The pre-processing included several filters, such as genes with fewer than 5 counts, cells with fewer than 200 expressed genes, genes present in fewer than 3 cells, genes with a high total count percentage, genes starting with MT, cells with N genes by counts greater than 200, mitochondrial gene counts less than 35%, removal of overexpressed genes, and selection of highly variable genes. Data normalization was then performed. The following steps were executed using Python and RStudio. Clustering was performed with ScanP, and cellular annotation was done using the Allen Mouse Brain Atlas and available literature. 2 pseudoreplicas were generated using pseudobulk analysis, and differentially expressed genes in the gliovascular unit were identified using DESeq2 and EDGER in RStudio, with a threshold of log 2 FC absolute value greater than 0.58. With the differentially expressed genes in the gliovascular unit, a functional enrichment analysis was conducted to identify lipid metabolism-related processes. A dot plot was then generated to visualize the expression of key lipid metabolism-associated genes and transcription factors. Our results are represented in 5 figures. In Figure 1, we show the clustering of different cell types in the hypothalamus of 3xDG, AD, and control mice. We observe that astrocytes and microglia are the most abundant cell types. Within microglia, we identified 2 subpopulations, disease-associated microglia, or DAM, and proliferative-associated microglia, or PM. In Figure 2, we compare cell type proportions and analyze differentially expressed genes in each cell type. DAM is more represented in controls, while PAM is prominent in the 3xDG AD model, suggesting its association with proliferation in pathological conditions. PAM microglia, along with endothelial and ependymal cells, show a high number of DEGs, indicating their potential role in the early lipid metabolism alterations in Alzheimer's. Figure 3 shows the functional enrichment analysis performed using the differentially regulated genes. We found that lipid signaling processes are highly represented in all cell clusters, with PAM microglia being particularly active in these processes, potentially indicating involvement in disease progression. Additionally, processes involving endothelial and ependymal cells are relevant, possibly due to their proximity to the blood-brain barrier and the strategic role of the hypothalamus in the brain. In Figure 4, we see a dot plot of the expression of key genes, including SLC27A1 associated with fatty acid uptake, Axel 6 involved in fatty acid activation. DHCR24 essential for cholesterol biosynthesis. BOT2 involved in phospholipid remodeling, and SEPT1 critical for phospholipid synthesis and membrane integrity. These genes showed specific changes in clusters like PAM and endothelial cells, reinforcing their importance in lipid dysregulation in Alzheimer's. Finally, in Figure 5, we present key transcription factors regulating these lipid metabolism-associated genes. We identified SEPA, AHR, ZEB1, and TAL1 as primary transcription factors due to their NES values and the number of target genes each regulates. These factors are associated with specific functions such as fatty acid synthesis, lipid homeostasis, transport, and storage. This suggests that these transcription factors could be potential therapeutic targets to restore lipid balance in the affected brain. In conclusion, our single-cell transcriptome analysis of the hypothalamus in 4-month-old 3xTG AD mice revealed significant changes in lipid metabolism genes, indicating their role in early Alzheimer's. Astrocytes and PAM microglia, the predominant cell types, showed high lipid signaling activity, with PAM microglia notably proliferating in 3xTG-AD. Key genes such as SLC27A1, AXL6, DHCR24, BOT2, and SEPT1, involved in lipid uptake, activation, and remodeling, showed expression changes, highlighting their role in early lipid dysregulation. Transcription factors SEPA, AHR, ZEB1, and TAL1, which regulate these genes, present potential therapeutic targets for restoring lipid balance in early Alzheimer's. Thank you very